Stakeholders watching this video are likely already convinced about the need to transition to more sustainable, equitable, and resilient protein supply systems. Our role as Wageningen University Research is to provide science-based evidence and technological innovations to support what we call the protein transition. Protein transition requires coordination between supply and demand. Production side changes should go hand in hand with societal changes in our consumption patterns. Now we're going to focus on the production side and talk about how we can reduce the European protein deficit, which is 20 million tons per year. We can reduce that deficit by increasing our production of non-animal proteins here in the Netherlands.
So the first source is pulses. Pulses are seeds of legumes harvested as dry grains, and they include different species like dry beans, dry peas, chickpeas, lupins, and they are actually very interesting crops for Europe for different reasons. One of them is that they are able to fixate atmospheric nitrogen, and this way precluding the use of nitrogen fertilizers. And the other reason is that they produce high amounts of proteins of good quality for food and for feed. So I think in a perspective of a protein transition, I would choose for going for a set of different protein crops, including pulses. From the pulses, I think that promising for the Netherlands would include faba bean for the very high yields, peas, as we know more about this pulse than others, and at a medium term I would also see very promising in lupins, given its very high quality of protein as well as low in starch. In the past decades, there have been only a few efforts to develop better varieties of protein crops. And if you want to enter a protein transition, you do need to have better sources and more sources of proteins from plants. And to that extent, at Wageningen University Research, we are working in a multidisciplinary group to develop these new varieties. And we combine expertise from the technologists, process technologists, as well as from the plant breeding to develop new varieties that lack these off flavors that will be generated during processing.
So the second source category of interest are agricultural side streams. The Netherlands is already a world leader in this field. AWB, for example, are cultivating potatoes and selling their proteins across the globe. Potatoes used to be cultivated for their starch, but nowadays the business case rests on the proteins. Another example are sugar beet leaf proteins. So recently, Kosen took over a patent from Wageningen University Research to extract proteins from sugar beet leaves, and they're hoping to bring that technology to market within the coming couple of years. And we don't want to stop there. We think there are interesting proteins in a lot of agricultural residues, including from tomato, cucumber, potato, even lettuce, from the stems, from the leaves, from even possibly the roots. These proteins are fundamentally different than the plant storage proteins that are available right now. So they deliver unique nutritional and functional properties. In Wageningen, we've done a small feasibility study to show that we can extract proteins from these materials, and our next step is to breed the crops for more efficient extraction of those kind of proteins and to help scale it up to a larger scale. We really believe that these proteins are interesting because they are basically already grown. We don't need new land or new resources to cultivate them. We just need to take those side streams and extract the proteins and make them available.
The third category of protein production that we work on in Wageningen is the microbial production. And the microbial production of protein is a very resource-efficient way of producing protein. For example, we can use phototrophic, light-dependent microorganisms like blue-green algae and microalgae that can fix carbon dioxide and nitrogen from the atmosphere and convert that into protein. Disadvantage of this approach is that protein is produced in a very dilute form. So we need extensive downstream processing and purification to obtain our final protein. Another way we can use this efficient production system of protein with these microorganisms is to use genetically modified microorganisms. We can actually make them produce animal-derived or animal-identical proteins like milk protein and like gelatin, and produce them in an efficient way. Disadvantage is that we have to deal with a lot of legislation in Europe, but also in the rest of the world, which inhibits application of this in food systems at the moment. A new way of using microbial fermentation for protein production is by using agri-food side streams or waste streams into protein, by selecting microorganisms that are producing the right composition of protein and the right amount of protein. This is by far the most efficient way of using microbial fermentation for human protein production.
In Wageningen, we are deeply committed to finding answers together. We want to invite all the stakeholders to engage with us and to contact us with any questions or comments. We're happy to be inspired by other people's visions. We're happy to think together about the challenges that our stakeholders are facing. And we want to end with an invitation to all of the stakeholders to reach out and connect and to help us create the future together.